RNA-Seq, the go-to technology for gene expression profiling. It lets us look at various types of RNA that's transcribed in an organism. This can be used to gain insight on a biological process or a disease state, like cancer, for example. In the 1970s, scientists Howard Temin and David Baltimore simultaneously discovered the enzyme reverse transcriptase. This enzyme generates complementary DNA from an RNA template. This is particularly useful because DNA is much more stable than RNA. Consequentially, DNA sequencing technologies like dideoxy chain termination, RT-qPCR, and probe hybridized microarrays, which pave the path for high-throughput DNA sequencing, can be used for RNA detection. There are many variants of RNA-Seq which modify the method in order to select for specific types of RNA, control for biases, or provide additional temporal and/or spatial information. The general workflow is relatively standard. It consists of the fragmentation of RNA molecules, conversion to cDNA by reverse transcription, high-throughput parallel sequencing, alignment to a reference genome, counting the reads, and then finally, analysis. So why do we care? First, RNA-Seq lets us identify gene isoforms which are generated through alternative splicing mechanisms. This allows us to see the different protein isoforms that are encoded for in a given gene. But that's just messenger RNA. There are other types of RNA in the body, such as microRNA, ribosomal RNA, short interfering RNA, and long non-coding RNA, which all have their unique functions. Thus, we have a relatively simple method which is generalizable to a family of biomolecules that can be used to understand biology. For example, differential expression analyses identify up or down-regulated transcripts between two groups. These groups can be stratified along different variables. A common one would be a wild-type or mutated version of a particular gene. Gene expression can also be used as a feature rather than an endpoint. Splitting patients according to the expression of a single gene is commonly done. You can also use multiple genes, which is the concept of a gene expression signature. There are many modern RNA-Seq methods which have improved upon the classic bulk approach. Single-cell RNA-Seq measures RNA transcripts of individually partitioned cells. This allows for identification of specific cell types in the sample based on their transcriptomic profiles. Slide-seq and GeoSeq build upon this by capturing spatial data to the detected transcripts prior to sequencing. This lets us know what's being expressed at a given tissue site. Live-seq uses fluidic force microscopy to take cytoplasmic samples from living cells prior to sequencing. This method doesn't actually kill the cells like the others do. Transcriptomic profiling is one of the tools scientists use to understand our biology, and RNA-Seq enables this. This was RNA-Seq in 100 seconds. Thank you for watching.